Thanks for joining us in the first talk in our fall seminar series through the RDAR Center. Uh, Without, I'm not going to belabor the point here, but just a few things, I guess, before I introduce the talk. There will be a Q&A at the end. We're going to do that through the Q&A feature of Zoom. The chat won't be working for that, and so we're going to do that just down at the bottom. You should see a little Q&A section there where you can submit your questions, and then at the end, we'll go through those. I'll read them out, and we'll do the whole thing. So just be aware of that. It's my pleasure to introduce Dr. Erik Garcia today, who's going to be, as you can see, giving a talk on hallucinogenic. I can talk today, I promise. Subhallucinogenic doses of 5-HT2A receptor agonists diminish opioid reinforcement, looking at sex and developmental implications. Just to introduce Dr. Garcia, Dr. Garcia is a behavioral neuroscientist and neuropharmacologist at the University of Nebraska in Omaha. His research identifies individual-level predictors that govern the development of substance use disorders. His latest research focuses on opioid use disorder and novel medication development within a behavioral economic framework to rapidly characterize therapeutic candidates. Dr. Garcia aims to bridge the gap between preclinical discoveries and meaningful clinical outcomes to aid people in their path towards substance use recovery. Most importantly, he is a firm believer and advocate for diversity, inclusion, and the mentoring of scientists. And I have a note here that he's also an aficionado at brewing beer at home. So I
That
would
is
love
very
to hear
true.
about that later. I might submit my own little Q&A just about that
Yeah.
later, because I find that interesting. And just a brief description of the talk today. Opioid use continues to be a major public health concern. The use of translationally relevant models of substance use can reveal the behavioral and neurobiological mechanisms that lead to uncontrolled consumption and overvaluation of drugs. This presentation will discuss the latest developments in identifying a non-opioid receptor target to suppress opioid reward and opioid value. Dr. Garcia's latest research indicates that the serotonin 5-HT2A receptor is a viable target, but this may depend on biological sex and early life developmental environments. And without further ado, I'll turn it over to Dr. Garcia. Thank you.
Awesome. Thank you very much, Dr. Barrett. I'm really excited to be here and sharing some of my lab's data. And really, I took this opportunity to try to highlight some of the work from my research team because they are in the lab doing the grinding and the running of animals. So hopefully we can get them highlighted today. And then, of course, happy to answer questions at the very end. To start, I have no conflicts of interest that would potentially influence any of the research designs or the analytical approaches or potentially any of the interpretation of the data. Okay, so why we're all here is to better understand not only the neurobiology of substance use, but then try to develop strategies, whether that's therapeutics or behavioral interventions that can ultimately try, they can ultimately try to help people who are struggling with this debilitating disorder. As we all know, I'm not going to belabor the point. The development of a substance use disorder is nonlinear. It can be a cyclical process encompassing both drug taking and binging and relapse-like episodes that can last for many, many years or perhaps even a lifetime. Those modifications seem to be associated with alterations in various circuits throughout the brain that have been extensively reviewed by George Koob and Nora Volkow over a number of different high-impact papers. While I think that's important, one thing that we know is that the medications for opioid use disorder, despite everything that we understand with the neurobiology associated with opioid use disorder, is lacking the medications to actually help. We know that when people are compliant with their medication, the outcomes seem to be good, but unfortunately, mu-opioid receptor antagonists often have poor patient compliance, and for physicians who may not be aware, they can actually exacerbate withdrawal symptoms if they are taken prior to a prolonged detoxification. So what this tells us is we need rapidly deployable medications needed for treatment And one of the things that I think that we can do is try to capitalize on receptor systems and neurotransmitter systems that might be outside of the status quo, so to speak. And one of those happens to be serotonin. Serotonin is near and dear to my heart. And why it is because less than 1% of all of the neurons in our brain are capable of synthesizing serotonin. Most of that happens within the dorsal median raphe, which sends ascending projections throughout the cortex. And really, the diversity of the signaling, which is what I always tell my students, and hopefully something that you kind of take a piece away today, is that really the intelligence is at the receptors. And the serotonin system is the most diverse GPCR family that we know of right now. There are 14 receptors. 13 of them happen to be G protein coupled receptors. And while all of them play their respective roles in mood and physiological arousal and mental health with anxiety and depression, what we tend to not focus on is their role in motivated behaviors. Now, more recently, the serotonin 2 receptors, particularly the serotonin 2A and 2C receptors, have been implicated in reward through psychostimulants such as cocaine and methamphetamine. And then more recently, within the last handful of years, looking at oxycodone reinforcement as well. Now, interestingly, some of those off-target effects that we see, I mean, the diversity of this family, you can kind of guess that drugs that might interact with serotonin 2A receptors might also impact serotonin 2C receptors, or maybe even serotonin 4, 6, 7, or 1A, right? It's very complicated to try to get that molecule to sit right in that binding pocket and have a preferential effect with a very limited side effect profile. The side effect profile has been very well documented, particularly as it relates to serotonin 2B agonists, because we know that chronic activation of those induces cardiac valvulopathy. And then, as we all know, maybe why you're here today, maybe serotonin 2A receptors have seen kind of this resurgence and as an opportunity to have a different kind of approach to treating psychological and psychiatric disorders, but traditionally many of them have been avoided because they are hallucinogenic. If you ask patients, many of them want the benefits of a hallucinogenic drug without the 4 to 12-hour psychedelic trip. That's pretty well established. These receptors all signal through what we call a serotonin or signal through metabotropic receptors. The serotonin 2 receptors kind of preferentially use the GQ phospholipase C pathway, which we'll get to a little bit later on in the talk. But ultimately, one of the strategies that people commonly take when they're trying to develop and identify is to repurpose already FDA-approved drugs. And I'm going to go through this relatively quickly because this is really some older data from postdoc, but it kind of helps set the story a little bit. And ultimately, Dr. Marcy Bubar discovered that serotonin 2C receptors are actually co-localized on dopaminergic neurons in the VTA. And that led to the hypothesis that perhaps turning on or activating those receptors could actually blunt opioid reward. There was a paper that followed that from Dr. Catherine Cunningham's lab that demonstrated that serotonin 2C agonism via lorcaserin was actually able to suppress oxycodone self-administration and what we would call oxycodone Q reactivity. We wanted to replicate this in a more translationally relevant point of view using a behavioral economics point of framework. And we generally hypothesized that pretreatment with lorcaserin would diminish opioid reinforcement by both decreasing consumption at a minimal cost, and I'll define these characteristics a little bit later as they become a little bit more relevant to the actual interpretation of the data, but just trying to get through this backstory a little bit. Long story short, we thought an increase in demand elasticity, which actually tells us that animal is getting a little bit more flexible in its behavior and it's starting to value that drug reinforcer a little bit less. So what we actually discovered is when we dose-dependently administered lorcaserin before a oxycodone self-administration session, we saw exactly that, a reduction in Q naught, while the animals seemed to be more flexible in their behavior, which was a really promising sign and provided some rationale or a behavioral mechanism as to how lorcaserin was actually reducing oxycodone reinforcement. These are the parameters that get extracted. So Q naught here is really interpreted as a consumption at a minimal price, or basically if it's as close to free as we can measure. And then elasticity really describes the slope, and that has been associated with the essential value or the reinforcement value associated with that. Now, it's inversely related to that. So actually, we want increases because that tells us that those animals are getting more elastic. Now, not to belabor the point here, but we wanted to understand really what the specificity of this effect was. So we used a selective serotonin 2C antagonist to try to block the effects of lorcaserin. And you can see here that when SB is pretreated with lorcaserin in this green curve here, those animals, you get rid of the effect that lorcaserin has to suppress oxycodone self-administration. So it appears as if this effect is mainly driven by the serotonin 2C receptor. However, and these are the same plots here, basically summarizing where the effect may lie. Now, interestingly, there was a translational discrepancy, which is what we struggle with as translational scientists to move things from the cells to the animals, ultimately to the bedside, to try to understand where this effect might be failing, right? So in human clinical pharmacology experiments, 10 milligram tablets failed to reduce cocaine and oxycodone self-administration, or really, if you ask people what are the subjective effects of the drug, they are very much still there. So something is different happening with animal and with a human. You could chalk that up to population variables, or where the people did have varying severities of cocaine use or opioid use disorder, et cetera. But if you take a more simplistic approach to understanding what the drug is actually doing on a pharmacodynamic point of view, you have to ask the question, is it really lorcaserin as selective for 2C over 2A? And I'm not so sure it is. Data from molecular pharmacology and just functional assay signaling suggests that it's only about 3, maybe 10-fold different. And if you're working or you know anything about in vivo pharmacology, that's not very much of a difference. So we kind of started to hypothesize at this time that maybe lorcaserin is actually functioning as a dual agonist of serotonin 2C and 2A, especially when you started to look at the signaling assays, whether it's a human serotonin 2A receptor or a mouse serotonin 2A receptor. This green line here demonstrates that lorcaserin is functioning as an agonist in this GQ pathway. So that tells us that on some level, maybe at those higher doses where we're seeing those therapeutic effects, that maybe lorcaserin is actually hitting 2A. So in order to test that hypothesis, the next logical step would be to administer an antagonist. And there is one called M100, M100-907, administered before lorcaserin, and it gives kind of this partial blockade, suggesting that at least some of the effect that lorcaserin has on demand elasticity to kind of elevate it, which is what we want a reduction in reinforcer value, is partly driven by serotonin 2A agonism. So that was somewhat of a surprise, and that really prompted these next series of experiments. I know I went through that really fast, and I'm happy to take emails or further questions if you have any after the fact. But that really prompted this next series of experiments, because what this suggested was that lorcaserin was actually functioning as either a full agonist or a partial agonist at those serotonin 2C and serotonin 2A receptors respectively. However, we know that lorcaserin had some issues pop up about 3 or 4 years ago, concerns about increased instances of cancer, as well as it being a Schedule IV drug, and really limits its further clinical use. However, with what we discovered about the potential activity with the serotonin 2A receptor, I became real interested in probing that effect to see, could we possibly identify the serotonin 2A receptor as a promising target to reduce reinforcer pathologies in general without having much of the hallucinogenic effects. So our entire goal here is not to make hallucinogenic trips and have them be therapeutic. I'm interested in asking the question, can therapeutics or hallucinogenics be therapeutics without necessarily inducing a hallucinogenic effect? Okay. So as with all animal pharmacologists, you eventually start with the food self-administration. And interestingly, serotonin 2 receptors modulate food reinforcement. Consumption and overconsumption of highly palatable foods is obviously associated with poor health outcomes, such as high blood pressure, cardiac disease, excessive weight gain, etc., and earlier instances of mortality. However, the motivational components of palatable food consumption are associated with reward regions of the brain. We know that food and food cues that are associated with highly palatable food activate the mesocortical limbic pathway, which is kind of our final common pathway that we think of when we think of drugs of abuse. And interestingly, serotonin 2A is expressed in that pathway, particularly in the nucleus accumbens and many different regions along the cortex, suggesting that it could modulate food intake. So what we had, or the research question that we were really after was, can serotonin 2A agonists change food self-administration? So I had a really promising undergraduate who eventually joined my lab as a graduate student now. He took 64 rats. 32 of them were going to self-administer sucrose pellets. The other 32 are going to self-administer high-fat food pellets, moving from a fixed ratio 1 schedule all the way to a fixed ratio 5 until they were stable. And importantly, these animals were not food deprived at the time of testing. They were all freely fed. And importantly, food reinforcement was paired with that discrete cue light, which was this flashing light that would tell the animal that they had earned a sucrose or a high-fat food pellet. Again, these were between subjects groups. So again, once stable, we pre-treated the animals with DOI at these selected doses and then observed them in a 30-minute food self-administration session. Over here on the left, we have pellets earned. I'm just breaking this data down really quickly, not looking at the actives. We didn't see any effects on the inactive lever presses, but how much sucrose pellets are these animals actually eating? What we discovered was that upon increasing doses of DOI, we saw a linear decrease in the amount of sucrose pellets that those animals earned. It did not depend on biological sex, which was interesting to us because we have females that are actually much, much smaller. Second to that, when we asked the animals or a different cohort of animals, well, what about high-fat food self-administration? We saw a very similar effect, except we saw males have a little bit stronger of effect, suggesting that DOI was more potent when we were talking about high-fat food self-administration. So these males were affected at a lower dose. So we felt pretty confident here that we had figured out a dose range that was not going to be disruptive to the animals, that they were going to go into the box and immediately start head twitching or have any kind of adverse effects. So this was the dose range that we chose to go forward with as we started to uncover and discover whether or not serotonin 2A agonists could be potentially useful for opioid self-administration. So as we all know, fentanyl overdoses are a continued public health emergency. We saw a 7.5-fold increase from 2015 to 2021. And alarmingly, if we look at the year-over-year data, it doesn't look like it's getting any better. In fact, it's plateaued. It's peaked and plateaued is what I should say, with well over 100,000 overdose deaths attributed to drugs in general, and usually about 70-ish% of those are attributable directly to opioids, which obviously is a major problem. So I had another promising undergraduate want to probe this effect alongside me, and she was a fantastic student in my lab. She's now at the University or Michigan State University, and trying to uncover whether or not there was any therapeutic utility to using serotonin 2A agonists as potential treatment for opioid use disorder. Again, historically speaking, many of these compounds have been avoided because of their hallucinogenic potential, and as we all know, many are still under Schedule 1 status. However, that resurgence in research prompted us to, there might be some justification here. Research from Lindsay Cameron out of David Olson's group has taken taken ibogaine and chemically modified it to create TBG, which we're looking at it as a partial agonist of the serotonin 2A receptor. It doesn't give a full agonist response, and it's also shifted to the right. So potentially as low potent partial agonist was TBG, was their molecule. But interestingly, when they took that and asked animals if we pre-treat them with TBG, it reduces heroin intake, which was really fascinating to us and really, again, provided some of that justification. So again, that molecule that they predicted is not going to be hallucinogenic. So we wanted to ask the question, could we use those sub-hallucinogenic doses, I'm sorry, to potentially reduce the reinforcing value of fentanyl? So again, how we're going to do this, how are we going to measure the strength of a reinforcer? Well, that's traditionally done using progressive ratio schedules of reinforcement. You can look at concurrent schedules of reinforcement, but I think what is really a hot take right now is understanding these relationships from a behavioral economics point of view. These pop up on a translational, have a translational impact. You can ask people how much they'd be willing to pay for one cigarette. Will they pay 50 cents? Will they pay $5? And consequently, we have developed animal models that you can ask an animal how much they are willing to pay. So the consumptions of goods is a function of price. So when the price is low, we would predict from just simple demand theory that consumption would be high and stable, which would be kind of represented over here. So if the unit price is pretty low, this is taken from Strickland and Lacey, you would predict that it would basically be kind of flat because those animals are going to be inflexible or inelastic on their behavior because the price is low. But as soon as we start to get to prices that are exorbitantly high, we should start to have some individual differences that start to predict this bend and this slope in the amount of the commodity or the consumption that's happening. So hopefully we can develop therapeutics that starts to bend this curve down and to the left, because what that does is it tells us that the animal's consumption is somewhat under the control of price. And if the price gets exorbitantly high, the animals will stop self-administering. So what we decided to do was test this molecule, again, DOI, because we had established a dose using the food self-administration, and looked at it because it is a preferential 2A agonist. It's a little bit cleaner than the other hallucinogenic compounds, although it does have some action at serotonin 2C as well. And what we decided to do was, again, use that same behavioral economics paradigm and use exponentiated demand curve fits using nonlinear regression and mixed effects modeling. Again, all the code that would be freely available, if you want this, please feel free to email me, and I can send it through GitHub or whatever you would need. But this is the analysis we chose to do, mainly because it handles the zero values a little bit better than the traditional Hirsch exponential equation. Okay, so what this looks like, in every session, a rat is going to get an 11-point dose response curve that's going to resemble something like this for each rat. And what this dose is doing is dropping by 1/4 log every 10 minutes in this assay, which produces doses that progressively drop all the way down to the very end of 0.0178. And if we transform that to price, how many responses or how many infusions does it take to equal 1 microgram, you'll see that the prices progressively increase. Price and consumption at each one of these bins are both transformed and then plotted on this curve. Again, happy to share code as you see fit. Okay, so males and females, we took an N of 29, and this paper was just recently published by Alice in Drug and Alcohol Dependence. Long story short, males and females readily acquire fentanyl self-administration. We just kept them on an FR1. There was no sex difference that we observed in number of days it took to meet acquisition criteria, which was defined as at least 10 infusions for 3 consecutive days, a greater than 2 to 1 active to inactive ratio, and less than 15% variability across those days. Okay, so we do get some changes because we're having such low sample sizes towards the end. So we'll all kind of look at the figures like that. Okay, so what about our hypothesis? DOI, once we give that pretreatment, there is traditionally a load-up period that the animals will do to try to get to their optimal brain concentration before they kind of do the demand task. What we saw was DOI does not alter the fentanyl load-up for male rats or female rats, suggesting that that initial brain concentration is not modulated by serotonin 2A activity. So that was a little bit surprising to us, but when we actually conducted the demand analysis, this is where we saw the interesting effects. So the serotonin 2A agonist, DOI, dose-dependently suppresses the reinforcing value of fentanyl. And what I mean by that is we start to see that curve bend down and to the left only for the male rats, whereas Q naught was seen to be depressed in the females. Now that's an interesting take because, and again, here's our summary parameters of what this actually looked like. We have the suppression of Q naught in the females, a slight elevation of Q naught in the males, but that effect was actually really small. What we're taking that to mean is that because there was no change in elasticity for the females, we're not doing anything to the actual reinforcing value of the drug. And instead, we might be potentiating that effect so that they need less drug to kind of maintain that optimal brain concentration. So at high doses or those low prices, we might actually be potentiating in the female rats the effects of fentanyl. Which obviously would not be ideal. We took a look at all of the extra parameters. How long did it take them to press? Were there any inactive lever presses differently? No, we didn't really see any of that, suggesting that we basically don't think we're causing behavioral disruption at the doses that we're testing. Again, we're using relatively small doses of our hallucinogenic drug to produce those changes in fentanyl value. So in our translationally relevant preclinical model, we determined that DOI suppresses the reinforcing value as evidenced by that elevation in elasticity. So we're bending that curve a little faster. And we're not having that same effect in females, which is interesting. There was opportunities for obviously to make the experiment better by taking menstrual cycle samples or trying to correlate those things. But honestly, we counterbalance the drug order. So any potential effect of menstrual cycle is probably going to be washed into all drug treatments for the females. So we're still trying to dig into how that might change DOI's effect for the females. Secondly, we revealed that for males, serotonin 2A is at least a novel opioid strategy that bidirectionally alters reward. Now, we think that we are using sub-hallucinogenic doses. I actually cut this out of the presentation just in the interest of time. But we do have the dose response for DOI in a head twitch assay task, which I'll talk about a little bit later. And we are well below doses that reliably induce a head twitch response. So we think we are using sub-hallucinogenic doses of DOI to kind of get these therapeutic effects. Now, the big question that we want to ask now is, okay, that's for fentanyl. Most people don't usually start taking opioids, start off with fentanyl. We have to ask the question, well, is this specific to fentanyl or does this generalize possibly to other opioids? So my current graduate student, Emily Koffenberg, has really taken on this project and asked the question, does this effect generalize to other mu-opioid analgesics that carry significant abuse potential? So these data are taken from the Substance Abuse and Mental Health Services Administration survey that usually publishes the data every October or November. And what you can see here is that the percent of people who are age 12 and older who are misusing are usually misusing hydrocodone and oxycodone. Interestingly, Caitlin Vanderweel has demonstrated that oxycodone induces a longer and sustained dopamine release inside the nucleus accumbens, so that efflux has been associated with elevated abuse liability. So we decided to focus on oxycodone to see whether or not DOI's therapeutic effects generalize to other opioid analgesics. And what Emily decided to do for this project was to, again, catheterize rats, train them to stable FR3, and then move them to our behavioral economics tasks and test them using basically the exact same procedures, except we're going to decrease by a quarter log. Oh, I think I have a typo. Every 15 minutes, not 20 minutes. I apologize. So here's the doses. So again, they'll sample in our low price twice economy, that high dose twice, because this first bin, they're going to have a load up, and we don't want to use that to estimate their consumption at a null cost. And then we'll sample again. So at this load up phase, is there any effect of DOI or, again, that serotonin 2A agonist on the load up? With our hypothesis being, again, if we should see it, it should be a decrease or a linear decrease. And again, we don't see any strong effect when it comes to oxycodone load up in these short access rats. So here on the left, green, we have increasing doses of DOI in our males. And in this, I don't know, pink, purple, and magenta color, I don't know what that is, we have no effect, right? So again, we're not decreasing the initial amount of drug that it takes to get to that satiation point, but now we have to ask the question, are we doing anything to the actual reinforcing value of the drug? Okay. Here, again, we have our demand curves. This is a little bit difficult to see on this computer, but this is the dark green curve is signified by the asterisk here with the alpha. And what we're seeing here is kind of that same pattern of effect, where we have increasing doses of DOI, and again, are tested in the same range that we tested with fentanyl. And instead, we're getting a nice elevation in demand elasticity, telling us that we're removing the reinforcing value of oxycodone for male rats. Now, the surprising thing for us happened on the right side of this figure, which is all of the hues of pink here. Because if you remember with the fentanyl, we didn't see a strong effect with alpha or Q naught. And instead, we are seeing that with oxycodone, which was really, it was a good sign for us, telling us that we're actually diminishing the reinforcing value in fentanyl as well. And it may not take such a high dose, which is what we thought initially with the fentanyl. Now, to further kind of validate whether or not we're actually measuring something that's translationally relevant, long access self-administration models compulsive intake, or you can use intermittent access. But long access self-administration drives elevated consumptions. It increases and prolongs drug concentrations in the brain, and it has been associated with decreases in opioid receptor expression, at least in the dorsal striatum. Now, we wanted to ask the question, is this a recreational model that we're seeing, or a recreational effect that we're seeing with our models? Or if we actually utilize a more compulsive-like drug-taking model, do we see a same pattern of effect when it comes to DOS? So really trying to validate whether or not serotonin 2A or low hallucinogenic drugs, do they have efficacy or not? So again, as you guessed it, we put rats through the same behavioral economic task. These are a different cohort of rats. There's no effect on the initial load up, which is, again, interesting to us. We're not doing anything that changes that initial load up. But what we are changing is how these animals respond once they start getting challenged with increasing prices of our behavioral economics task. Over here, again, a little bit difficult to see, at least on my computer, so I apologize for that. Dark green is the Q naught, so we're decreasing Q naught. And in the light green, which is this curve, I'm going to highlight it for you, this curve right here, we're actually getting that downward bend into the left that we would desire, suggesting that we are decreasing the reinforcing effects, but only at the 0.01 dose of DOI. And instead, at this dose of DOI here at 0.1, we're probably potentiating the effects of fentanyl, which is a little bit counterintuitive, but I think that we have an explanation for it. Over here, for females, we're getting the desired effect that we want. We get a drop in Q naught and a significant elevation in alpha, telling us that we are decreasing the reinforcing value of oxycodone for our female rats. Now, this effect over here, again, is interesting because it suggests that after prolonged long-access oxycodone self-administration, what we have is a potentiation of 2A signaling upon a challenge with DOI. When you have higher doses of DOI, it's actually pushing that curve more leftward and making fentanyl more potent. So DOI, just to summarize this section really quick, DOI reduces fentanyl reinforcement in males, but not females. DOI reduces oxycodone reinforcement value in males and females, but that depends critically on the long access component. Did these animals actually self-administer a lot of drug over a 6-hour time period? If it does, you might actually shift that DOI response curve a full log over. Okay, so our highest dose of DOI potentiated oxycodone reinforcement. Now, mu-opioid receptor downregulation or desensitization, which is what we're thinking is probably happening with long access self-administration as you start to see those escalations in tolerance, may actually enhance serotonin 2A signaling. And this is really the backbone of this work came from Erik and George Agonists, who demonstrated that there was significant interactions between mu-opioid receptors and serotonin 2A receptors, perhaps in the cortex. So next, we decided to really test whether or not this interaction between mu-opioid receptors and serotonin 2A receptors was actually relevant. So we could use what's called a head twitch response assay. Now, the head twitch response assay distinguishes how it's traditionally used. Hallucinogenic from non-hallucinogenic 2A agonists. So animals will head twitch after they get a serotonin 2A drug that is hallucinogenic. So it can be used, we think, as a behavioral proxy to understand whether or not a new compound has potential hallucinogenic effects. So this is data taken from Adam Halberstadt, who has very nicely demonstrated that drug discrimination correlates really nicely with head twitch responses, suggesting that the intraceptive stimulus effects of hallucinogenic drugs are tightly correlated with the head twitch response. Second to that, recently, work from Javier Gonzalez Meso as well as Alana Jaster has really demonstrated that there are significant sex differences as it relates to head twitch response, and biological sex may play a role in that as well. And we're seeing those kinds of same effects when we're talking about the hallucinogenic profile and how they could be used as a therapeutic for opioid use. So what we decided to do here was really innovated by another one of my undergraduate students, Jade Escarcega, who decided to use serotonin hallucinogenics after mu-opioid receptor agonists, so getting them dependent on it. And where this really came from was that full agonists and partial agonists suppress DOI head twitch responses, such that when they are on board, DOI has less of an effect. And as I mentioned, George Agajanian, mu-opioid receptor activation reduces those excitatory postsynaptic currents. So all of this to say, we think that there is probably a G protein coupled receptor interaction happening between mu-opioid receptors and serotonin 2A receptors. And if we induce a dependent and tolerant animal with chronic escalating doses, we might be able to downregulate this receptor and in a behavioral pharmacology way, stimulate it with a dose of DOI. And if we see enhanced DOI head twitch responses, that's at least one indication on a behavioral pharmacology level that these receptors are actually interacting with each other. So chronic opioid exposure will downregulate those mu-opioid receptors, and we should see an elevation. We kind of hypothesized that females would be more susceptible to this potency shift because they are more susceptible to the head twitches, according to Dr. Jaster. Okay, so what we did here, I'm going to flash forward here and just get to the timeline because I think it's easier to follow, is they arrived here. We gave them injections of oxycodone or saline in a completely between subjects design. We gradually escalated that dose over a number of 10 days. And then on the 11th day, or I guess day total 18, we challenged them with saline or DOI. with, again, our hypothesis being that the animals who had previous oxycodone exposure would have less mu-opioid receptor governing serotonin 2A receptor activity. Now, while we did see an effective DOI or a main effective DOI, suggesting that DOI was working, it was doing its thing, we didn't see really a potency shift or a change between the saline exposed and the oxycodone exposed rats. We initially saw, and then when we fully powered out the experiment, some of these effects, well, mainly that guy, was driving the lack of effect there. But interestingly, we see another sex difference, and that is there is a strong potency shift in DOI in animal and female rats who had previous exposure to oxycodone. So that was surprising. That kind of supports our hypothesis. I'm going to jump forward here because this is the time course analysis if anybody was interested to when these effects were happening. But the story remains the same. Female rats who were exposed to oxycodone had a longer and more sustained interaction. Now, the data that we are really excited about right now is the intracellular signaling component of this, because as I started off this lecture, or this seminar, I should say, is we think that G protein coupled receptors interact with each other and we can capitalize on those G protein signaling pathways. Ultimately, we are kind of thinking from the behavioral evidence that we have probably upon DOI stimulation, greater serotonin 2A activity. However, the kind of inverse hypothesis that we need to test is, well, what if under native conditions we have serotonin 2A? What if we add mu-opioid receptors to the cells? Do we get a dampened signaling to kind of test the other side of this hypothesis? So serotonin 2A receptors, as I said, signal through the GQ pathway, ultimately results in an intracellular release of calcium, and that's going to be our readout going forward. So I'm not going to belabor the point here because we can chat later if there's questions, but we titrated DNA concentrations for either mu-opioid receptor or serotonin 2A receptor and kept them the same, whether that was the full receptor or whether there was a kind of a control vector carrying the red fluorescent protein on either side. Now, we did a calcium mobilization assay to, again, measure the readout and measure them on different tools, as well as a GI readout that I think I'm going to skip over for right now. So what does this actually look like? Again, data that we're really excited about. In our kind of positive control condition, we have serotonin 2A getting expressed with the red fluorescent protein. That red fluorescent protein really shouldn't do anything to signaling. As we would predict, as you increase the concentration of DOI, you get a stronger effect compared to lower concentrations of DOI. Cool. Under mu-opioid receptor and the kind of negative control condition with a red fluorescent protein, we have the same DOI compound, but DOI is not an agonist at the mu-opioid receptor, which is important for our hypothesis because if DOI was just bleeding over onto the mu-opioid receptor, well, mu-opioid receptors couple to GI, and that would actually explain the dampened signaling. But we don't see any effect even when we test out to 10 micromolar. Now, the interesting side here. When we have serotonin 2A co-expressed with mu-opioid receptors, even though we are at saturating concentrations of 10 micromolar, et cetera, we have a dampened signaling somewhere hovering around 60 to 70%. So when we plot the full concentration response curve with the serotonin 2A receptor signaling with DOI, and the serotonin 2A receptor that is now sharing that space with the mu-opioid receptor, we get a downward shift in our concentration response curve, hovering around, again, 60-ish% of the actual signaling, which fits with what we're seeing on a behavioral pharmacology level that kind of sees both sides of the coin with that hypothesis, that when they're together, mu-opioid receptors are actually blunting that signaling and may explain some of that potency shift that we saw when we saw the long access self-administration experiment. Okay, so I know I'm running close on time here, I think. So summaries, in vivo and in vitro pharmacology indicates that serotonin 2A probably have a functional interaction with each other, and that they reveal that the serotonin 2A may sex-dependently be a novel opioid strategy. I think we still need further examination into the safety and efficacy profile of chronic use of these drugs, but it's a good start. The therapeutic potential may depend, importantly, on the plasticity of the mu-opioid receptor as after chronic opioid administration, whether that's oxycodone or fentanyl. So our future studies are really aimed at trying to elucidate some of those effects, deciding or deciphering, whether or not it's cell type specific and what part of the brain is really where we're at right now with this project. I wouldn't be where I am right now without incredible support from a variety of people, including my family, Chloe, even though my daughter and son fight like cats and dogs, love them to death. Funding, the RDAR pilot grant, which has been tremendous in getting me off the ground in my faculty position, a grant from the Peter McManus Charitable Trust, Conda, et cetera. Tremendous help in my first couple of years. And most importantly, the undergraduate research team, which most of this data has come from. They are a great, fantastic group of students, highly recommend, and curious and keeps me young. So without getting too soft over here, I appreciate everything you all do, and I can't wait to continue working with you.